Types of viruses. There are 3 types of viruses on the basis of type of host. A. Animal viruses. These viruses cause diseases in animals. B. Plant viruses. These viruses cause diseases in plants. C. Bacterial viruses or bacteriophages. These viruses cause diseases in bacteria. Classification of viruses. 1. On the basis of nucleic acids. Phylum, Vira. Subphylum, Deoxyvira, DNA viruses. I. Class Deoxyhelica, helical symmetry. Order, Chitovirals. 2. Class Deoxycubica, cubical symmetry. Order, Haplovirals. 3. Class Deoxybanala, virus with head and tail. Order, Urovirals. Subphylum, Ribovira, RNA viruses. I. Class Ribolica, helical symmetry. Order, Radavirals. Order, Sagovirals. 2. Class Ribocubica, cubical symmetry. Order, Gymnovirals. Order, Togovirals. 2. On the basis of envelope. A. Enveloped viruses. These viruses have envelope outside the capsid. B. Non-enveloped viruses. These viruses do not have envelope outside the capsid. Reproduction of viruses or viral replication. There are following steps in viral replication. 1. Attachment. 2. Penetration. 3. Biosynthesis. 4. Assembly. 5. Release. 1. Attachment. The attachment of the phage to the surface of a susceptible bacterium by means of its tail is called adsorption. Host specificity of the phage is determined in the adsorption stage of the cycle itself. Artificial injection by direct injection of phage DNA can be achieved, even in strains of bacteria that are not susceptible to the phage. The infection of a bacterium by the naked phage nucleic acid is known as transfection. 2. Penetration. The process of penetration resembles injection through a syringe. The phage DNA is injected into the bacterial cell through the hollow core. After penetration, the empty head and the tail of the phage remain outside the bacterium as the shell. 3. Biosynthesis. During this stage, synthesis of bacterial protein, DNA, and RNA ceases. On the other hand, phage DNA, head protein and tail protein are synthesized separately in the bacterial cell. The DNA is compactly packaged inside the polyhedron head and finally the tail. Structures are added. 4. Assembly. The assembly of phage components into mature infectious phage particle is known as maturation. 5. Release. Release of phages typically takes place by the lysis of the bacterial cell. During the replication of phages, the bacterial cell wall is weakened and it assumes a spherical shape and finally burst or lyse. Mature daughter phages are released. Significance of viruses. Viruses are a kind of biological puzzle to biologists since they are at the threshold of living and non-living things, showing the characteristics of both. Viruses are very much used as biological research tools due to the simplicity of structure and rapid multiplication. They are widely used in research, especially in the field of molecular biology, genetic engineering, medicine, and are utilized in production of vaccines, used to develop immunity against viral infections, etc. Viruses are used in eradicating harmful pests like insects. Thus they are used in biological control programs. Phages are used as scavengers to eradicate bacteria present in polluted water. Viruses have living and non-living properties, so they are important in determining origin of life.